Welcome, everyone, and thank you for joining us for the Black Alumni of MIT Research Slam. I am Tisha Turner, MBA02 BAMIC Programming Chair, and I'm delighted to welcome you here today to spotlight some of the up-and-coming best and brightest Black researchers from MIT. This event is in partnership with the MIT Alumni Association and the Office of the Institute Community Equity Offices at MIT. I'm joined by our judges, Keandria Bickerstaff, class of '89, president of KenQuest, Dr. Deborah Carlton, class of '75, physician specialist in internal medicine, and attorney Aidan Allen, class of '02, of the law firm Wilson, Sonsini, Goodrich, and Rosati. Judges, could you warm us up by telling us where you're calling from, what your past year has been like, and maybe what you're most looking forward to this evening? Keandria?
Hello, folks. I'm calling you from Austin, Texas. It has been a very busy year. I've been very fortunate that even with pandemic and closings, my business has been good. I work in the area of intellectual property at this point in my career with patents and for law firms and for corporations. And it's gone very well. And I'm looking forward to being able to grow my business now that we can get out a bit more and attend more conferences and business opportunities. So I'm optimistic about this year.
Keandria. Deborah?
Deborah, you're muted.
There we go. I'm sorry. And I hope I don't lose my internet again. We had a power surge, so hopefully we're stable now. I'm Deborah Carlton, class of '75, that is 1975, course 7. I live in Atlanta, Georgia, where, as you heard, I'm an internal medicine physician. I, in fact, retired from that area of my life in 2020. But I've had an excellent 2021 and going into 2022. It's been busy with both studying and training. I have become board certified in a second specialty, the area of lifestyle medicine, which is a scientifically proven approach to preventing, halting, and in some cases, even reversing the chronic illnesses that we see ravaging our communities and the country. Examples of those illnesses are type 2 diabetes, cardiovascular disease, high blood pressure, obesity, and many cancers. The 6 research-based pillars of lifestyle medicine are a whole food, plant predominant eating pattern, regular physical activity, stress management, avoiding risky substances, and positive social connections, along with restorative sleep. So hopefully your interest is piqued and you'll go to the American College of Lifestyle Medicine website, lifestylemedicine.org, to learn more about how to help yourselves and your families and friends and colleagues. Also, I'm a triathlete and I'm training for a longer race than usual this year. I'm training for a half Ironman in September. So that's taking up a lot of my time too. So I'm really happy to be here, be a judge, looking forward to hearing what our researchers have to say. It's a challenge communicating complex materials, and I'm hoping to take some ideas home for myself. So thanks, everyone.
Thank you, Deborah. And last but not least, attorney Aidan Allen.
Thank you, Tisha. Hi, everyone. I'm Aidan Allen. Like Kay, I'm also in Austin, Texas. This last year, we're almost done, but been homeschooling 2 of my kids, fully homeschooling due to COVID. Can't wait for them to be done in about a month now. It's been sort of a challenge. It's been challenging as president of 2 organizations here in Austin, the Austin IP Law Association, as well as the Austin Black Lawyers Association. And it was somewhat challenging trying to make sure everybody stayed connected throughout the pandemic when we couldn't really get together. And so I'm definitely looking forward to things opening up as we go forward, still knowing that I have very, very young kids and have to be somewhat cautious. Tonight, I'm looking forward just to hearing from our budding researchers and things like that, just finding out what people are working on and just really hope to take away something that just is, it just not necessarily blows me away, but just really just creates a thought in my mind, just like, wow, I hope that is going to get to market, or that's going to be successful and obviously be a research or resource later on if anybody has any questions and any thoughts about, how do you move things forward from where you're at in this initial phase of just research and just thinking of what to do. So I'm looking forward to this. Thank you, BAMIT, again for inviting me to judge this. So thank you.
Thank you, Aiden. And thank you to all of the judges so much for taking the time to help us with this event. We know you all have demands on your time, and we appreciate it. I do not envy the work in front of you, but I know you will have fun. With that, let's get to the main event. In the next hour, we'll hear from 6 alumni researchers. We'll ask them all to come on video now so you can see them. Please help me give a warm welcome to Janelle Fouché, Stuart Isaacs, Jasmine Joseph Chazan, Quiley Simeon, Thela Townsend, and Herbert Turner. We will be posting a list of all the competitors and their affiliations in the chat. Now, here are the rules of the event. Our competitors will present their research in 5 minutes or less. They are allowed to use 1 visual slide. We'll ring a subtle bell when time is up, and 10 seconds later, we'll make less subtle sounds. Judges will evaluate research pitches, not the research itself, using the following criteria. Did the presenter make effective use of time, explain the topic in terms understandable to everyone, speak clearly and at an appropriate pace, capture your attention from the beginning of the presentation and hold it, motivate or advance propose viable solutions to the problem, question, or goal, convince you of the importance of their research, create an effective visual that supported the pitch. After each pitch is completed, we will field 1 question from the judges. Please use the Q&A feature on your Zoom toolbar for questions. We will try to get to them when the judges are deliberating after the presentations. Our competitors will take home glory, publicity, and some good tips on communicating their science. But there's also cash prizes at stake today. A total of $2,300 thanks to our friends at the MIT Alumni Association and the Office of the Institute Community Equity Officer. Now we turn to our presenters. In alphabetical order by last name, we will start with Janelle, then Stuart, Jasmine, Quiley, Thela, and then Herbert. Good luck.
Good afternoon or good evening, depending on where you're calling in from. My name is Janelle Fouché, she, her. I was at MIT from 2007 to 2011, where I studied economics and management science at Sloan. Did a few Europs in econ and knew I wanted to get a PhD in either economics or some econ related field. So that's how I ended up at Harvard. I'm now a G5 studying education policy and program evaluation. And this is one of the few papers that I'm currently working on. It is co-authored with a professor at BU who used to be at Harvard. Long story. Okay, so without further ado, my dog thinks this is the best time to be up under me. All righty. To move, nope, I don't know where to move this. Oh, Okay, so this paper is in part motivated by the news. So I put in 2 articles from the Boston Globe. On the left, you see a story about Troy Martin, who was a senior at Amesbury High School, which is about an hour north of Cambridge, and his snowmobile hit a snowbank and he sort of died on impact. The other article is about Raekwon Brown, who's a 17-year-old gunned down outside of his school in Dorchester. And the reason I chose these 2 articles is to show you, this is a phenomenon that is like plaguing some schools more than others. B, sort of how the headlines vary depending on the race and scenario of the student. But C, to also highlight that aside from like the core hard sciences, social scientists are trying to understand how these like terrible tragedies and phenomena impacts kids in school and beyond. So this paper is motivated by the widespread interest in how trauma, loss, violence, what have you, plays in the perpetuation of economic and racial inequalities in educational outcomes. I'm going to keep clicking. Okay, great. So the core research questions for this paper are, what are the death statistics for school-age children in Massachusetts public schools? Are there any differences in death rates or exposure to peer death by race, gender, and or income? And lastly, what impact do students' deaths have on their peers' educational attainment? This paper sort of fits in among some of the other econometric papers that try to look at loss and trauma in its various ways and how that impacts sort of the racial achievement gap, the socioeconomic achievement gap, and a host of other interesting things that we find really interesting, and I hope you do as well. To answer these 3 questions on the left of the 2 columns here, we built a novel data set that combines data from the Massachusetts Department of Elementary and Secondary Education and the Registry of Vital Records, that's a subsidiary under the Massachusetts Department of Health. So the first sort of like results that I'd like to show is a descriptive chart, and it looks here at the rates of peer death among K-12 kids. So as you can see, the biggest disparity, I guess this is where my mouse is, the biggest disparity that I noticed in sort of building this paper is that Black boys have a higher death rate than white and Hispanic boys. There's less of a disparity between Black girls and white and Hispanic girls, but one sort of still does exist. And so in thinking about the core research questions, right, here we have it. Here are some of the death statistics. There are clearly differences by race and gender. I'm going to leave aside some of the socioeconomic stuff for now, since I want to show you some of the causal pieces. This is another figure that we made that sort of looks at whether or not you've ever been exposed to a peer death. So in my grade, do I know someone that passed away? Really depressing stuff, but I do think it's really interesting, especially for the implications. These are some of the impacts that we found on high school graduation. So if you're exposed in the 9th grade, you're associated with a 5 percentage points lower likelihood of graduation. And that number sort of goes down over time. Folks in 12th grade less likely to be as affected by it, but still affected. These are some of the point statistics that we ran when you sort of look at it for Black, Hispanic, white, low income, and non-low income. There continues to be a disparity between Black and Hispanic versus white, and between low income and non-low income students. Lastly, the way kids die really matters. And I thought that's something that's really interesting to point out, with homicide sort of having the biggest point estimate on this like graduation and educational attainment outcome. So that is where I will end. Thank you guys so much. I hope that was only 5 minutes.
Yep, you didn't hear the sound, so you were well within time. Thank you very much, Janelle.
Thank you, and apologies for you get it.
No problem. Now it's time for a question from the judge. I'll turn it over to Aiden for his question.
Yes, thank you, Janelle. That was appreciate the slides and understand the technical difficulties. Main question is, I know there's a lot of research, but what is your overall goal in in studying this, and how in the real world do you see this actually playing out? Or how do you sort of foresee taking this research to the next step for in the real world?
Oh, absolutely. Thank you for that question. I think as a policy sort of minded student, I'm always like, okay, what's the implications? What do we do with this information? I think there's two main things, right? One, what is it that kids in non-low income schools or high income schools are doing right, when this sort of tragedy occurs, right? Are they hiring more grief counselors? Are they hiring therapists? Are they giving everybody A's? What are they doing differently for kids to bounce back quicker or better than kids in low income schools doing? So that's number one. And number two, it's determining what the mechanisms are that are causing this lower graduation rate, right? So is it a bereavement effect where kids are just so distraught by losing a friend or a peer that they're like, oh, I'm done, I can't carry on, school's too much for me, I quit? Is it some sort of short run effect where we're looking at like a trauma and stress related reaction to like seeing, a peer one day and then all of a sudden they're not there anymore? Or is it recognizing that like we too are mortals, mere mortals? And if you're in a case where, homicides are really prevalent in your neighborhood, is there a way to sort of problem solve with local leaders on how we can like avoid and like make school zones safe places and safe havens so kids can just focus on their school and not be traumatized by what's happening in their neighborhood? So I'd say there's still a lot more work for me to do on terms of potential mechanisms. But I think we found something really interesting.
Thank you.
Thank you. Thank you, Janelle, and thank you for that question, Aiden. Now we will move on to our next presenter. That will be Stuart.
Let's do this. All right, whenever you're ready, I'll go ahead and start. Okay, I think I'm gonna start now.
Go ahead.
So my story begins in rural Burkina Faso in West Africa. As an undergraduate, I worked on a team to help my friend and colleague, Birima Kabre, create locally built chicken egg incubators that are solar powered. As someone who had worked with the revolutionary Thomas Sankara, Birima strongly believes in communities being able to feed themselves and have self-determination. And he also believes solar systems can enable that. And it's not just Birima. Communities throughout Africa are looking to off-grid and mini-grid solar projects to meet their energy needs since they're currently being unmet. Of the estimated 650 million people that will lack electricity in 2030, 85% of them will be in sub-Saharan Africa. And in Burkina Faso, for example, only 19% of the population had access to electricity in 2016. In rural areas, this number is even lower at 5%. Many believe that solar can help turn this around, but there's a problem. During our field tests in Burkina Faso, our prototype totally failed. Our data was showing that we were getting way less power than we had predicted. Now, at first, I just assumed I had made a mistake in the design, but then I saw an MIT news article on air pollution impacts on solar panels. And I asked myself, what if all the dust I had seen was reducing the power output of our panels? This became the topic of my current research. In my work, collaborating with satellite engineers from NASA JPL, I aim to quantify the impact of dust on solar panels in West Africa so that we can better account for it in the design of our solar systems. Now, dust can reduce the power output of panels in a couple ways. First, it can reduce the amount of sunlight reaching the panel when it floats in the air as an aerosol. The dust particles will absorb and scatter light before it can reach the panel surface, reducing the power output. Second, it can collect on the glass surface of the panels, like in this picture, which also reduces the amount of sunlight reaching the energy converting material underneath. In my work, we use physics-based models based on literature methods to estimate how much dust impacts solar power output by using environmental data. This data comes from three main sources. First, we use very high quality ground-based sensors that provide us ground truth values. But unfortunately, there are very few of these in sub-Saharan Africa, so this is not enough. Second, we use a data set called MERA-2. It's essentially a model that imports data from things like satellites and ground-based sensors to make estimates of the values that we need. It has a great time resolution. However, it has poor spatial resolution of about 40 miles by 40 miles. So lastly, we use satellite data from collaborators at Washington University in St. Louis, or WUSTL, improving the spatial resolution by more than 5 times when compared to MERA-2, but with much worse time resolution. By using the data, we first looked at the role of dust as aerosols. We measured the amount of aerosols in the atmosphere by using a parameter called aerosol optical depth, also known as AOD. And for context, at an AOD value of 2, only 14% of the incoming sunlight will reach the ground. Immediately, we saw that the harmattan was significant. The harmattan is the annual dry season in West Africa when dry desert air from the Sahara Desert blows dust into the region. And in Burkina Faso, we can see that daily values as high as 3 during the harmattan. Here we have a chart showing the AOD at monthly average values in Burkina Faso. This is far bigger than anything we've seen in the US, where the average AOD value is 0.15. And we believe that this is why data sources like MERA-2 and the satellites don't capture the region's highest AOD values. These technologies simply were not designed with this region nor this application in mind. Now, although MERA-2 and WUSTL may not capture the highest values of dust, they are still useful for our work. For starters, we found that a discrepancy between the ground truth and the MERA-2 data could lead to solar panel undersizing by more than 10%. Additionally, we can use the data to understand regional patterns. For example, we saw that the maximum soiling losses for uncleaned panels will occur near the Bedell Depression in Chad, one of the largest sources of dust aerosols in the world. Ultimately, my work quantifies the amount of losses due to dust. We can see in Beni Zimbu, Niger, there can be up to 15% reduction in solar power output annually due to dust combined effects. Using our results, my friend Burima can properly size his panels and can now produce working solar power incubators in Burkina Faso. But I want us to go back to the first picture. Burima is standing in front of dusty solar panels because no one bothered to clean it. And that's because they don't work. I would argue that dust isn't the only issue here. A European NGO had installed these solar panels, but when a component broke, no one in the area knew how to fix it. The reason that our solar power incubators had to be locally built in Burkina Faso was so that local people can maintain it, but also because imported incubators from Europe aren't made to work in this environment. What if this concept also applies to the solar panels? In the future, I want to use our results to evaluate different panel shapes and materials that might be more fit for operating in this region. By innovating alongside local communities, I hope that our work will aid in Burima's goal of self-determination using solar power. Thank you, and I'm happy to answer your questions.
Thank you, Stuart.
Hi,
And now,
Stuart.
okay, Keandria, yeah, go ahead.
Hi, Stuart. This was a great presentation. Thank you so much. Question for you. Have you been able to give an estimate of how much dust in the air or in the atmosphere, the impact of it separate from dust that would be on the panels themselves? Have you been able to say, oh, it's a 20% factor versus a 50% factor?
Yeah, great question. So we do that. So we use the aerosol optical depth data to understand the dust aerosol impact. And you can see that actually here in this chart in the dark blue. So we can see that in this particular location, we're getting about 7.5% of annual losses due to just dust as aerosols. The other half we can estimate based on a model we use to predict how much dust is going to collect on the panels, assuming we don't clean it, assuming it has a certain angle. And we can see that over the course of a year, that's going to also add another about 7% to the losses each year. There's a lot of assumptions baked into that, but generally we can use this model to understand the different contributions of the different parts of dust reducing power output. Does that answer your question?
Definitely. Thank you so much.
No worries. Thank you.
Thank you again, Stuart, and thank you for the question, Keandria. Now we'll move to our third participant. Our third competitor is Jasmine.
Hey everyone, give me a sec while I set up the screen. Can you guys see that okay?
Yes, we can.
Okay, great. So hi, my name's Jasmine. I was course 20 at MIT. I graduated in 2018. For those of you who aren't familiar with the course numbering system, that's biological engineering. Now I'm a PhD student in the immunology program at Harvard Medical School, doing my dissertation work in the lab of Dr. Alex Shalik. So while my presentation today isn't on my dissertation work, it is a project that I've been a part of since joining the lab in late 2020. and is a large collaboration between many industry, clinical, and academic partners across 3 different countries as well. So the main goal of this project is to develop a better understanding of the immunologic landscape of chronic HBV or hepatitis B virus infection in the liver, with an eye towards development of proper treatment regimens for those suffering from this disease. So by way of background, HBV or hepatitis B virus is a non-cytopathic, meaning that it can propagate without killing the host cells, virus that infects hepatocytes or liver cells, and those are these cells here. Though it's not cytopathic, the burden of HBV virions can actually trigger an immune response, inflammation, and even cell death absent of immune responses. And persistent infection can ultimately lead to cirrhosis and hepatocellular carcinoma. Of the several types of HBV that we know about, of the several types of hepatitis viruses that we know about, HBV actually contributes the most in terms of hepatocellular carcinoma and cirrhosis, as well as acute hepatitis, leading eventually to death, which is why it's very important for us to want to better understand what's going on in the landscape of this disease. According to the WHO, also, almost 1/3 of the world's population has already been infected by HBV at some point in their life, and meaning that there's a great need to understand the pathogenesis of this virus and plan proper interventional strategies. Currently, the main strategies are prophylactic. So we get HBV vaccines in most countries, but they're not effective in patients who are already chronically infected. Treatment options are mostly antivirals. And while these will curb viral load to being below detection limits of normal assays, complete elimination hasn't yet been made possible, leaving patients still vulnerable to developing resistance to those drugs, hepatocellular carcinoma, and really needing to be lifelong patients on drugs in continuity. So because of the multicellular space that this virus infects in the liver, we really wanted to take an approach to kind of deconvolute the system and break it into as small components as possible. And this is something that my lab really focuses on. We utilize single cell transcriptomics to go from complex cellular mixtures such as this to single cell transcriptomes, which utilize the RNA. If you remember from biology, intro biology, we go from DNA to RNA to proteins. So RNA is the middle step that can tell us what a cell is doing at any given moment in time. And then from that information, we can tease apart different cellular characteristics and whatever cells are present. We can even do this with viruses too. And then there are different strategies for doing this. My lab mainly uses picowells, but you can dissociate your complex cellular mixtures and process the cells in a multitude of ways. So with this, there's another complication. For us to be able to analyze the liver space, we need tissue biopsies. However, tissue biopsies are really invasive for the patients. And just because someone has a chronic disease doesn't mean they necessarily need to or want to go under the knife repetitively so that we can understand what's going on in their system. So efforts have been made to see if peripheral blood could be a surrogate for tissue biopsy. And unfortunately, we don't see the same cell types represented in peripheral blood that we do in tissue biopsy. A workaround for this is fine needle aspirate biopsy, which utilizes a thin hollow needle that is inserted into the abdomen and basically sucks up little bits of the tissue in a much less invasive manner. This is a much better way to get essentially a small tissue biopsy without having to put someone under. So the beginning stages of our project focused on comparing fine needle aspirates and peripheral blood collection cell type representations across different transcriptomic processing pathways. We compared single cell RNA sequencing using picowells and droplet-based systems to do an analysis. And in the beginning stages of that, we saw that depending on which processing protocol you use, you'll get different cell types represented to different degrees. And I know that's very vague. And unfortunately, I won't have time to go into very many specifics with that. But essentially, they both have good coverage of diversity, but some more than others. And so the conclusion of this was that we would proceed with the picowell-based system, which our lab already specializes in, so that worked out really well for us. And going forward, we would continue to use that system to then start to ask questions that would be really important to understanding the biology of HBV, including questions of why do some people progress from chronic hepatitis to functional cure while others do not. Thank you.
Thank you, Jasmine. And now you'll have a question from our Judge Deborah. Go ahead, Deborah.
Thank you, Jasmine. That was very, very interesting. It's a complex topic. My question is, what do you think your research can lead to in the clinical realm, specifically in the case of chronic hepatitis B?
Sure, yeah. So I think that the main question we really want to find an answer to is if there are specific immune components that we find in certain patients that might not be in other patients that really allows them to progress from this stage here. It's the fourth panel where they kind of oscillate between controlling and not controlling the virus until they finally go to functional cure. So we want to understand what components of the immune system are allowing people to cross that threshold and control it, and if there's something that's preventing them from crossing over that hump, how can we address that? And we've partnered with industry collaborators such as Janssen, and I know that they definitely want to develop a drug that could help there. So that's kind of the main goal of this.
Great, thank you.
For sure, thank you.
Thank you, Jasmine, and thank you, Deborah, for the question. Now we move to our next presenter, and that will be Quiley. You're on mute.
Can I just ask how the previous presenter got their presenter view? The previous person that had this problem. And I could do a new share.
So one way around it is if you share, you can go into advanced and click a specific region that you want to share. Or you can do like that, yeah.
I'm gonna do new share. Yes. Okay, can you guys see me?
Yep.
You see my slide? Okay, awesome. So like I introduced myself, I'm Quiley, and today I'll be actually talking about an independent project. So I'm a PhD student in MIT Brain and Cognitive Sciences, but this is an independent project of mine because I'm really interested in bioinformatics and this like area combining AI and biology. So many of us probably know what DNA is to varying levels. You probably know of DNA as the molecular blueprint of all life. From high school biology, you probably also heard of the central dogma that DNA gets transcribed into RNA, which is then translated into protein, as you can see in this diagram. Now, protein has gotten a lot of attention lately, and more so recently with the breakthrough in DeepMind's AlphaFold, which is said to like solve this protein structure prediction problem. And Often we hear that structure guides function in biology, and we often hear this in the context of proteins. But did you know that actually RNAs, there are many RNAs that are not translated into proteins, and they have very rich structure which guides their function. And these RNAs are called non-coding RNAs. So not just proteins, it's not just proteins in which like the structure is important. Actually, RNAs have very rich structure, and that guides their function. And we actually don't know a lot of the function, but the architecture of structured RNAs is very hierarchical. So you start out with a base sequence or a sequence of bases, and those base pair in a certain way, and that gives rise to the secondary structure, which you can think of as the 2D structure. And there are certain features in this 2D structure that you can like annotate, such as helices and stem loops, pseudoknots, and those are really important for their function. Now, in this talk, I'm gonna focus on 2D structure. For the majority of RNA molecules, the secondary structure is highly important to the correct function of the RNA. For example, 3 general types of RNA structure are double helix, stem loops, and pseudoknots, and many others that are annotated here in this figure. Now, in the past few years, we've actually been able to sequence a lot of RNAs that we previously didn't know the function of, and many of which we still don't know the function of, because the technology is available to do so. And a lot of research now is focused on, given this primary sequence, which is a sequence of bases, can you predict secondary structure? And ultimately, our goal is to figure out what is the function of much of this RNA in our cells that we don't know what it does. And previously, people thought, oh, this is just junk RNA, it doesn't do anything. But what we're finding is that it actually has a lot of important functions. For example, maybe you know tRNA, which is like very important for actually making proteins. And that's an RNA, it's not a protein. So in this work, the hypothesis that I came up with is that probably RNAs with similar structure, and in this case, I'm dealing with secondary structure. I'm not going to do anything as advanced as DeepMind. They probably have similar functions. And I wanted to ask, can we cluster these non-coding RNAs whose sequence that we have based on their secondary structure? So what I did is I used some of the deep learning approaches that have been applied to this. So many deep learning approaches will take in the primary sequence and predict the 2D structure and give you a diagram such as the one you see, such as ones that you see on the left of this slide. So I extracted what we call embeddings of the 2D structure from a fully connected layer of a deep network that was trained strictly to predict the 2D structure. And this is just a visualization of what this looks like. So you can think of it as a high dimensional vector. And then I do something called dimensionality reduction, where you cluster this high dimensional, for example, imagine 1028 dimensional space, and you bring it down to 2 dimensions. And then I do this k-means clustering to identify clusters. And then I validate my results by looking at each cluster and seeing whether they actually clustered according to secondary sequence. And what you see here in these results that yes, like we can actually find that RNAs cluster according to their secondary structure. Now, why is this important? Well, like I said, for the majority of RNA molecules, their structure is really hierarchical. And nowadays, prediction of secondary structure from RNA primary sequence is a really intense area of research because we have all this RNA structure data and we have all these new methods based on deep learning that accelerate progress in this field. And I was really inspired by this idea that structure guides function in biology. So if we believe that the secondary structure is important for the function, then perhaps we can use this atlas of non-coding RNA structure similarity to give us ideas of functional similarity. So the future work that I'll talk about is actually focused on finding out whether these RNAs that are clustered based on structure, do they also cluster based on their function? And if we can figure out the function of RNAs, then we open up a whole new area of biology. We can open up
Riley, that's time.
And so I've used in future work, I intend to use a graphical neural network approach to compare the distance between these RNAs. And also future work will look at whether you can compare the distance between these graphical structures of RNAs. And that will help us to uncover new molecules. So to end, I'll just like to acknowledge my collaborators on this work, Annie Ruda, Waitse, and Tony, who helped me with this project. Thank you.
Thank you, Quiley. And now we'll have a question from the judge. Your judge will be Deborah. Go ahead, Deborah.
Thank you, Quiley. That was very interesting. So, so you're clustering these non-coding RNAs according to their structure. And then the next step is to kind of, how do you foresee that next step occurring where you try to figure out what their function is or if they have similar function? How do you see that working?
Yeah, so the idea is that there are very limited non-coding RNAs whose function that we know, but there are a few. So if you're able to find a neighbor in one of these clusters whose function is known from previous research or from a database, like there are lots of databases that annotate these functions, then you are, this allows you to generate hypotheses about the function of a particular RNA. But more importantly, I think what I really want to go for is it can help you generate hypotheses about particular motifs in RNA. So like particular substructures, like the pseudoknots that I showed, the multi-loops, like what are their particular role in the overall structure of a larger RNA? And that can help us to design, in a sense, designer molecules, which you could imagine could be very useful for drug research and drug development.
Thank you.
Thank you.
Thank you, Quiley. Thank you,
Sorry.
Deborah.
I'd like to point out, I didn't quite hear the subtle sound, so I'm not sure if that was me or did anyone hear it?
No, we're having technical difficulties, so it'll be me. I'll let you know when it's time. But thank you. Now we'll move on to our next presenter, and that will be Fila.
Hi, everyone. Again, my name is Fila Townsend. I am a PhD candidate at the Rutgers School of Management and Labor Relations and Sloan MBA 2013. Before I jump into my research, I just want to ground in something important, which is that I identify as a labor activist scholar. What that means is I study labor and employment systems and look at the working and living conditions of people who work. And I'm grounded in a justice and equity framework. And so I think that's just an important framing for my approach and how I look at the topic that I'm really excited to share with you all. So with that, I will share. Actually, you would think by 2022, okay, let me put in here. Actually, sorry. So I just tried to be really simple and straightforward with my slide here. The working title of my research, which happens to also be my dissertation research that I'm in the throes of currently doing field work, is What is a Black Worker Center? A Critical Exploration of Race and Class in Worker Organizing. So what I really explore and interrogate here is, what are these organizational forms called Black Worker Centers? What role do they play? Why do the groups of workers who organize them form them in the first place? And what is their role in a broader community, particularly communities who are focused on what it means to build power for themselves and to change their lives? Now, I'm sure you're asking, like many people when I talk to them about my research, which is, what is a worker center in the first place? Simply put, worker centers are nonprofit community organizations that support and advocate for workers, particularly those in marginalized communities. By marginalized, what I mean here, most workers that are connected to worker centers are in low-wage jobs. They are or underemployed. They really, literally and legally fall outside the boundaries, the rights and protections that labor law affords to many of those who are covered under it. So I won't get into a bit of labor history or sort of the limitations or nuances of labor law, but the point being is that worker centers have emerged as organizational forms to help support workers that are, again, in our most marginalized communities. And perhaps surprisingly or not surprisingly to many folks, most of these workers are of color, are immigrant, and women. So what I set out to explore here is how we understand these community organizations as sites where race and class, and I would add gender, sort of intersect to shape not only the lived experiences of the workers in the communities that they're connected to, but specifically and more central to my interest and topic is how these social forces of race, class, and gender, I would add, shape how these workers perceive and maneuver and orient themselves to build, whether it be economic or political or other social forms of power in their communities. Why this matters? Well, race and class and gender and these sort of social constructions of identity, if you will, what I've seen in my work, and there's a whole host of literature that would add to this or bolster this, people's racial and gender identity in particular actually shape how they think about and develop strategy and organizational forms. And that was interesting to me as I did some preliminary research where I talked to leaders of organizations and asked them, many of them unprovoked, offered evidence as to how their racial or gender identities meant something for how they oriented themselves around their work, how they thought about what power was in the first place. And for me, again, as an activist scholar who's really trying to explore and interrogate questions that I think matter for how we think about changing the world, understanding levers of power, how we maximize power, how we build more power to affect change in our lives is really critical, and I think lies at the heart of these issues. But we also know from literature and critical race theory, racial capitalism, even more critical forms of theory, whether it be Marxist or other things, that how people see and orient themselves around power actually shapes, again, the organizational forms that they choose to go into. Many folks who are maybe outside of political establishment or more mainstream establishment structures of society, the resources that they're able to leverage and access in the first place are different. And they have to be because of a function of their position in the political economy. So what I'm really trying to do in this research is look at this particular context of folks organizing marginalized workers who in this context happen to be Black workers, to think about how is it that they come together? Why do they come together? And really, why a Black worker center as opposed to maybe some other kind of organizational form? Such as a trade union, if it's about building economic or political power. Why not a church, which, as we know from civil rights movement of the 60s and 70s, was a really important place for community activism and advocacy? Why not pursue, and they do in some cases, but other pathways, higher education, which many of us have pursued, or some other economic or political development strategy?
Sheila, you're at time.
What is a Black worker center and how does it sit in this particular role as these workers come together and think about the best, most effective ways to build power? So that is what I'm interrogating, engaging in my field research now to better understand.
Thank you, Fila. And now your judge question will be coming from Aiden.
Thank you, Fila. Definitely appreciate the topic and the in-depth that you went into. I guess I'm more interested in how do you actually conduct your research in this area? How do you reach out? How do you sort of get the survey, the results that you're presenting, and sort of I'm more interested in sort of the background of, where do you get your data from? Yeah, your methodology here.
Yeah, so I largely engage in a variety of qualitative methods. So one method is interviews with the organizers and strategists of these centers. Again, really thinking about how structures shape their strategic choice and decision-making. I also am engaged in some bit of an ethnography where it's been a bit difficult with COVID, but to some extent being able to embed in structures with them and just be in the spaces and really see in real life what their strategies are like and how they unfold underground, and really try to, in talking to them, but also seeing the strategies and behaviors that they engage in. My work is really trying to uncover the mechanisms and the processes through which race, class, and gender dynamics actually show up in real life. So I'm really trying to bridge theory of what these literatures and theories say about these social constructions with how they actually unfold on the ground. So interview, ethnography, participant action research, to some extent, I'm there with them, maybe engaged, but I try to, you have to keep a bit of a distance as a researcher. So largely through those qualitative methods, I'm exploring and interrogating these questions.
Thank you.
Thank you, Fila, and thank you, Aiden, for the question. And now for our final presenter, Herbert.
Okay. All right, so can everyone hear me and see my screen?
Yes, we can.
All right. So with the growth of megaconstellations in space, satellite operators' biggest fear is no longer, where did my multi-million-dollar satellite go? It's, where did two of my multi-million-dollar satellites go at the same time? My name is Herbert Turner. I'm a senior at MIT studying aerospace engineering and computer science. And today I'm going to talk about my work on collision avoidance for satellite constellations. One of the most important measurements to take when worrying about collision avoidance is orbit determination of satellites. And the biggest source of error for low Earth orbit determination is inaccurate atmospheric density estimates, a layer of the atmosphere that affects low Earth orbit satellites. And current prediction methods for this area of the atmosphere are either very computationally expensive and slow, or not accurate enough for real-time prediction. So as part of the Space Systems Laboratory, I've been working on reduced order models for thermospheric density modeling. So what reduced order models do is that they try to approximate high fidelity models in a much lower prediction space. So as opposed to looking at 10 to the 4th to 10 to the 7th n-dimensional spaces, we look at n-dimensional space where n is 10 to 100. And this allows us to preserve the essential behavior and dominant effects that affect density modeling while being computationally fast. And so what we do is we take the original data, we reduce it to this 10 to 100 dimensional space, and we make our predictions there in real time before reconstructing our prediction in the original input space. And my work specifically was on using machine learning methodology to do this reduction, prediction, and reconstruction. So the data we looked at for this was grid point density data over many longitudes, latitudes, and altitudes from the years 1999 to 2008. And we took this data from two commonly available data sets, JB 2008 and NRL and MSIS, which are pretty well known in the field. And we used two types of models. The first was a convolutional autoencoder. So this convolutional autoencoder takes input data and compresses it to some embedded space before reconstructing it in the original space. And by carrying out this process, you train two important models: an encoder, which can map you from the input space to the embedded space, which we need to make our prediction, and then a decoder, which takes our output prediction and can map it back to the original input space. And then we also trained a deep neural network to make the prediction in the embedded space. So this model was trained so that for a given time, it would give you the density if you gave it solar weather data and the density data at the previous time step. So after we trained those models and spent some time tuning the parameters for them, we decided to look at our results. And so we evaluated our models based on the historical data from the previous data sets. So before training, we set aside part of this data that we then used in our evaluation. And so we looked at using our deep neural network for predictions versus what's called dynamic mode decomposition with control, which was the baseline model that the lab had used before I began my work. And we also looked at two different methods for doing our data reduction. The convolutional autoencoder that I talked about and the previous method from the lab, which is proper orthogonal decomposition. And we found that if we looked at our predictions over a 48-hour time period, that using this deep neural network over the baseline models of the lab meant an order of magnitude difference in the mean squared error of the density data. And we also found that if we used an autoencoder over the baseline method of POD for both the reduction and reconstruction of the data, we got 20% better results than with the baseline. And so with this results, we now have improved models that we can use to make thermospheric density data, which can then be fed into overall systems for orbit determination and collision avoidance, which allows satellite operators to operate much larger constellations of satellites with minimal collision risk.
Thank you, Herbert. Now a question from a judge, Keandria.
Thank you, Herbert. This is very interesting. How you said there was a 20% improvement over previous technique. This is a system that can be deployed in real time, or is it currently being deployed in real time?
So this sort of subsystem is part of a much larger system that the lab is working on. So basically, to do this orbit determination, there are a lot of variables that go in. And so thermospheric density estimates are one of them, but not the entire system. So yeah, this is sort of a smaller subsystem that can't be deployed on its own, but is useful for teams working on larger systems.
Excellent.
And so this is actually the issue that caused SpaceX to lose several satellites a month ago, is that the density changed a significant amount due to a solar weather storm. And so they had to deorbit a lot of satellites.
Yeah. Thank you very much.
Thank you, Herbert, and thank you, Keandria. Well, that concludes our presentations. Thank you, Janelle, Stuart, Jasmine, Phila, Quiley, and Herbie. I know I speak for all of us when I say that we are so proud of you. That was really impressive work that you're all working on. Also, we know that you have many priorities competing for your time, and we really appreciate your participation in this event. We're now going to give you a moment to catch your breath. While the judges tally their scores for 10 minutes or so, we will ask the audience participants today to weigh in on the crowd favorite. Judges are following the strictest of criteria on science communications to make their decisions, but this winning competitor will be an unfiltered Audience Choice Award to which we invite the crowd to apply all their favorite filters, whether they be on the topics presented or the style of presenters. We'll now make the poll live and give everyone some time to vote. Competitors, in the meantime, we'd like to get to know you a little better. In the same order in which we just heard from you, can you please tell us a little bit more about the context of your work? Describe the lab or department you're working in, the colleagues, environment, city or country, and your impressions of it, and perhaps your career ambitions beyond this work. We'll start off with Janelle.
Oh, so fun to always go first. Thank you. So if you could just repeat that list of questions real fast.
Sure. You could start off with a little context about your work.
Okay. Oh, okay. Yes. Okay. So I came into Harvard wanting to broadly look at education policies that, for lack of better terms, really just benefit Black kids. And originally sort of had like a first gen low income higher ed focus. And since coming here, I sort of fell in with the abolitionist crowd, and I've done a lot of teaching inside prisons and jails, and done some work on the school to prison pipeline, and seriously considering how to build out like the prison to school pipeline, and sort of helping people get back on track after they're released. So that's like where my passion is. Currently working with a professor at the Kennedy School on designing a curriculum that sort of focuses on the 13th documentary. I don't know if folks have seen that on Netflix, and having people contextualize that work. You know, how have they seen various criminal justice policies play out in their societies and in their worlds, and sort of like, what do we imagine public safety to look like in the future? So that's something I'm really excited about. In terms of research projects, you've seen the one here where we look at peer death exposure and various high school outcomes. I have another paper that sort of looks at the impact of restorative justice on a host of outcomes, and that's work with the Santa Ana School District. Another project that is now starting is looking at the impacts of non-prosecution. This is a really long title. Non-prosecution on like future criminal legal system involvement. So last year, some economists found that for the marginal defendant, actually prosecuting a misdemeanor violation actually led to future like criminal legal system involvement, and they didn't really know why. So we're undertaking interviewing sort of paired defendants, one who was prosecuted, one who wasn't, to sort of see how their lives diverged. And in the case of non-prosecution, it ended up being a better outcome for them, which again also has really huge implications for public safety and social policy. So I'll pause there in terms of projects that I'm working on and things I think about. In terms of what I want to do, if you're hiring, let me know. I think I'm supposed to say academia. I see the value in an academic career. I do love teaching. I do love doing research, but I think I want to be a little bit more policy focused, and you got to really establish yourself as a professor for a couple of years in order to jump into the policy world. So we'll see where I end up. I will pause there. Please tell me I answered everything.
You did. You did a great job. Thank you very much, Janelle. And now we'll hear from Stuart.
Okay, sure. Yeah, so I started thinking about this after my trip to Burkina Faso, and actually I wrote up the research proposal. I don't know if I'm spotlighted or not. So
You are.
oh, okay, great. Okay, so I wrote this research proposal in Professor Danielle Wood's course, like Space Enabled, looking at like space technologies for justice and development. And so the proposal came out pretty good. And so that's how it ended up getting funded by NASA. She had contacts there. They're looking for projects to work on. And so they're like, oh, this sounds cool. So we're doing it. And so that's been cool. Originally, my work started doing like alternative jet fuel for airplanes. So I'm in the aerospace engineering department, aeronautics and astronautics. And typically I look at using clean energy to turn water and air into fuel for airplanes, which sounds really cool, but it didn't feel that great in my spirit. So I left for a year. I went to Ghana. I taught for a little bit. And then like that really cemented this, like, oh, okay, this is like a real problem. I should probably do this. And so right now I'm looking to like try and like merge these topics by looking at basically using clean energy to make hydrogen for lots of things, but for African development, ideally, because that it feels good in my spirit. And I guess it also is relevant for the aerospace stuff, since that's the department I'm in. I think one of the people asked what motivates me to get up out of bed every day. I like knowing that communities I care about are like going to benefit from my work, or at least I'd like to think that they can. So that really motivates me to do the work every day.
Thank you, Stuart. Yeah, now Jasmine.
Hey everyone. So I guess context for where I am now. I mentioned my lab and such during the presentation. I'm an immunology PhD student at Harvard. I studied bioengineering in undergrad, and throughout undergrad, I kind of did a variety of different research projects. Like I worked in vaccine development for infectious diseases. I worked in cancer biotechnology. I did a sustainability and microbiology project in Ghana. And most of my research happened in other countries. So that kind of gave me it made me really interested in global health and global health equity. But I still wanted to keep a hand in the realm of engineering and being able to be in the lab. That's how I kind of settled on immunology as the topic for my graduate school. And the reason I wanted to go to graduate school in the first place is because I wanted to be able to know what it takes to be an independent researcher and to really see a project through from beginning to end, because that's something I felt like I didn't really get to do fully as a trainee in college. So that's where I am now. My lab in particular focuses on single cell transcriptomics, which utilizes RNA. I love that that was featured in 2 presentations today, RNA. Really, I like to think of it as a systems immunology place, and it really allows us to decipher different disease contexts, really whatever you're interested in. You can work on, and we really try to make sure that our work is doing some good in the world. So we have lots of partners in other countries, especially ones we do a lot of capacity creation. So we're trying to make sure that everyone can do the work that they want to do, no matter where they are. And then I guess going forward, I want to use my experiences to help. The things that I'm interested in, I guess, going forward are teaching and communication. I think communicating about science is really critical to society, especially when it comes to medicine and things like sustainability. So I don't know exactly what my profession would be or what my goal profession would be, but hopefully I can incorporate those 2 things into whatever it is.
Thank you, Jasmine. Wiley?
Hi, so I guess about me, I'm, like I said, a first-year PhD student. I'm an international student. I'm from the Caribbean in St. Lucia. The work I am interested in, I'm interested in computational neuroscience. So how do neurons, or how do the brains of humans like compute things that are so quintessential human, like learning, memory, emotions, and so forth. So I study those in like both computational models, so in silico stuff, as well as like simple animal models like C. elegans. In terms of the impact of my work, like besides basic science research, I really am interested in trying to find ways in which this can translate to, for example, biotechnology or bioinformatics, and help us to create new technologies that both complement those we have now, as well as like such as those we haven't seen. So an example of that is the way humans learn is very different from the way like current AI methods used to learn. So for example, by studying learning rules in biological networks, perhaps we can create new learning algorithms that can create even better AI and robots and so forth. So that's kind of the research that I did.
All right, thank you, Wiley. Now we'll turn it over to Fila.
Hi, everyone. So I don't know where to begin. I found my way into this work and research through, I think, a long career, as I mentioned, working in labor and employment relations, but at different positions. I've been in the private sector, I've been in the public sector, currently as a grad student in a union. In the last couple of years, while I've been pursuing my PhD, I've also done a fair amount of policy work, primarily at the intersection of tech and labor policy. But I think like within the last several years and everything that's been happening in the broader climate, the political instability we've seen, and really a lot of my work working alongside social justice and movement organization and worker advocacy organizations, this topic emerged. So there was a lot that I wasn't able to get into, which is really something that's happening in academia right now, particularly in spaces around work and employment, where people are seeing, and frankly, a bit late, coming to the game to understand how not just racial and gender and other identities shape the organizing decisions and strategies of workers, but also how the structures imposed on all of us through policies and laws in various ways also are shaped by social constructions, for lack of a better phrase, of race and class, et cetera. So my work is sort of positioned at this intersection to really understand how this particular group of workers, which in this case happen to be Black, although I think like this is work that is necessary to look in all contexts, but really thinking about how and why folks come together, build community, build solidarity, and what are the most effective strategies to do it. I think this is a time when, as I don't know if I mentioned, but like, you know, people are looking to institutions for guidance and finding it in different places in different ways. And it's happening in various ways, whether it be church, whether it be political organizations or institutions on various aisles, right, lines of the spectrum, whether it's more conservative or more liberal or whatever. And so for me, what I think is really interesting about these worker centers that focus on Black workers, and what's interesting, I didn't get to make this point, is that all of these people organizing in these centers, while they center and focus on Black workers, they actually see the centering of Black workers as instrumental in improving the living and working conditions for broader swaths of communities. So it's not a Black only type of strategy. It's seeing that the liberation of Black folks and Black workers and achieving better outcomes for them actually helps to move the needle for broader groups of people, right? So that's where, by happenstance, I've sort of landed in this work. I don't know what more to sort of offer and say. I guess in terms of future work, you know, I'm really interested in whether it be in an academic context or not, thinking about how we get more resources, money or otherwise, in the hands of folks that are organizing in their communities. So thinking whether it's around newer financial models, leveraging cryptocurrency, or thinking about other, again, financial or financing models. That's sort of the space I'm in right now. So I'll end there. If folks have any questions, feel free to reach out to me, but thank you.
Thank you, Fila. And now we'll hear from Herbert.
Hi, everyone. I'm Herbie again. Yeah, so I'm a double major in aerospace engineering and computer science, and I'll graduate this spring. And then I'll be doing a master's in computer science at MIT next year, just because I have no idea what I want to do after graduation yet. So I don't currently work with the lab that I presented my research with. I actually moved on to a lab in ocean engineering. And so I do a lot of marine robotics now. And I'd say I'm pretty excited about that work actually, because it's much more user focused, I guess, where working with small aquaculture farms to figure out how we can, use marine robotics to make their production more efficient, because labor costs are a lot of the prohibitive factors for letting them grow and be successful. And then, yeah, otherwise, I help lead 2 clubs at MIT, the AR/VR club and a new ocean engineering club focused on autonomous systems. So I guess my passion there is just education for younger students. So, teaching them different tools they can use for AR and VR, and then also just how to look at engineering problems and get started on solving them. Yeah, nothing else to add unless there's a question I missed or anything. I don't think there was anything specific to me.
Okay, thank you, Herbie. Well, thank all of you for sharing some additional insights about yourself. As you can see from the chat, everyone's very excited about the work that you're doing, and you will be hearing from several members of the audience after the event. But now I understand the judges are back. So I will, hold on. Should I take a few questions? I'll take a few questions from the audience and ask to the presenters. So one is a dual question, and I think I'll ask it to everyone. I think it approaches for everyone. The first part is how your research empowers you. And then the second part is, what will you do with your winnings? So I won't take it in the exact order as it was before, because I know Janelle wants a chance to not go first. So we'll start with Herbie this time. So Herbie, again, the question is, the 2 questions are, how does your research empower you? And what will you do with your winnings?
So I guess I'll talk about my current research for the first question. I think it's nice for a change to, be working with actual people across the country, and being able to talk with them and see how, my research will actually affect their lives, and seeing that up close and personal. That has been really empowering to me, to make me feel like, I've learned skills over the years that I can use in the future to help people and organizations. And then for my winnings, there's a charity working on water wells in Africa that my friend is doing a charity run for. So I'm hoping if I get the People's Choice Award, that's where that will go. And then I'm also on a trip right now. So I'll shamelessly say I also need gas money. So that's also where a good amount of it would go for any winnings I receive.
Thank you, Herbie. Fila?
Honestly, as boring as this is, probably helping to fund, if I win, my research, my field work that I'm engaged in right now. So yeah, that's how if I do win, that's what I would do with it.
And how would your research empower you?
Oh, I mean, I'm definitely, this is largely why also I'm interested in questions that require qualitative. I mean, I'm always empowered and motivated to actually be in community and working with people who are doing this kind of work. And so I think to not only see what they're doing and witness it, but also to more directly bring the learnings from research into real world spaces where you can see the impact is super rewarding and empowering. And so that's why I actually put a lot of my time and effort into operating, as I said, in this more activist scholar mode where, I'm able to help translate what I'm learning in my research to folks that are ready to put it into practice right away. And then, of course, in turn, I'm learning from them. And there really, in my opinion, should be a close-knit relationship between, obviously we want to make it objective and have sort of a healthy distance from it, but so much of our work and our view and perspective is needed in folks who are doing work on the ground in whatever space, whether it's policy, whether it's organizing or advocacy, or even in corporate settings. So I love being an interlocutor, a translator, someone who can help create spaces of synthesis where we're bringing ideas together and connecting dots to folks that really need it to advance the kind of work they're doing.
Thank you, Fila. Now we'll hear from Quiley.
Hi, so I think can you repeat the question? Is it what motivates?
Sure.
you?
How does your research empower you? And then what will you do with the winnings?
Yeah, so I think my research empowers me because it's not a well-trodden path. So I do feel like I have the opportunity to make a lot of impact in this field. It empowers me because it's something I've been interested in from a young age. Even before, like when I was applying to MIT, I was interested in sort of computational neuroscience as well as the applications of it. But if I try to explain to someone, it seems kind of far-fetched. But I've recently seen over the past few years, with the recent advancement that we've seen in both neuroscience and deep learning, computer science, it's actually a very emergent field with the possibility to sort of transform different industries. And I think it's empowering to be a part of that, especially as a Black student, as an underrepresented minority in academics in general, but even in particular in neuroscience and in particular in the subfield of neuroscience. So I think that's what motivates me, is that I can have a great impact and possibly also inspire other people like me to go into areas of science that are like new and very research driven. And I think how I would use my winnings is not that interesting. Probably I would use some of it to help with the expenses of my parents coming here for graduation this year, but I think I would just save half of it, put it into a IRA or something. So yeah, that's how I would use my winnings. Yeah.
Thank you. Now, Jasmine.
So as far as how it empowers me, I think on a very personal level, I am really trying to build my confidence as a researcher, and all of the failures that happen in the research lab, while they are momentarily shaking, really help to build resilience and help me to understand what it means to really follow a path all the way through, and then have experiences later on that I can share with others. So I think that that's really important and will empower me even if I don't know it right away. And then as far as winnings, if I were to win, I think that firstly, I'd want to provide lots of snacks for my mentees. I'm mentoring a couple different people, and I would love to like take them for coffee chats or just send them like a small care package or something. And then this is a shameless plug. I am one of several chairs of the PI reunion committee this year. So I want to send the rest of my committee care packages, and I think, but we don't really have the funds for that. So if I were to win, I think that I'd try to send them something like a little like pair of socks or something for all the hard work they're doing.
Thank you, Jasmine. Now we'll hear from Stuart.
Great. I think the first question had something to do with my research and how, I'm sorry, could you repeat that question? I'm so sorry.
No problem. How does your research empower you?
Got it. Yeah, I think this is one like it feels nice to do work that I actually care about. Like, I feel like I'm using my STEM skills in a way that I've kind of imagined I could, but I've never really had a chance to. So it's nice to like actually be doing it. And like, people are like, oh, this is like legitimate work. Like, that's kind of cool because I feel like I've always kind of been doing it on the side, or like trying to at least. So for me, it like is really validating to feel like this could actually be a career. So I like that feeling. And I'm like, oh, I can keep doing this potentially. That's nice. In terms of what I do with the winnings, I had some friends really give me a lot of help and feedback on the presentation. So I'd like to like throw some cash their way because they deserve it. Like if I won, I feel like it's because they helped me. And I think with the rest, honestly, my friends were recently like flaming me because my pots and pans are really old. So I might get some new pots and pans so I can like, I don't know, cook better food, I suppose. I have no idea what I do with the rest. If there's more to go by. I don't know how expensive pots and pans are. I think they can get quite pricey actually.
Thank you, Stuart. And finally, Janelle.
That was phenomenal. Thank you, Stuart. How does my research empower me? I think it's, and this is sort of tied to the like other question about like what gets you up or how do you get out of bed every day? Because research can be very long and laborious, and I've been working on one paper for at least 2 years. I think I'm just trying to stay focused on like the big picture. Like I read my personal statement a lot for why I like applied to grad school. And it's because I really, really care about Black and brown children, and I really care a lot about education. And so I think using my quant and qualitative skills and like just ways of like building community with folks for a better cause, for more just policy, what have you, is really empowering and really motivating. Even on the days that I really, really don't want to get out of bed. And I should say, I'm like literally overcoming COVID. So I was like nervous I wouldn't have a voice right now. So this is awesome. So really, I appreciate you guys for putting this on. And the winnings, it's probably gonna go to my credit card bills because I was Uber eating a lot when I was in quarantine. So I'll leave that alone. Yeah.
All right, thanks, Janelle. Thanks, everyone. Now, I do believe the judges have returned from their deliberations and they are ready to announce. So I'll turn it over to the judges.
Okay, I think I'll go first. I think each judge is going to announce the one winner for third, second, and first, and I'll start with third place. But before I do, I want to say thank you again to all of our presenters. Truly appreciate the time that you've taken to put together your presentations and to spend some time with us tonight to explain what your research has been and for the last couple of years for some of you. And so I definitely appreciate all of your time. And again, I think I say that on behalf of all the judges and everybody else who's watching. So with that said, I will like to announce that the third place is Jasmine Joseph Chazan, or Chazin, I'm not sure if I'm saying that right. Congratulations on that. We really liked your slides. We thought they were very well done. The one, I would just say personally, one of the things, it got a little small at times, and it got, it could have just been a little more focused on sort of the problem and start off with the problem first, which I think was the deaths. And you sort of went to that, I think, like second, in just sort of the order of how you get to catch the audience attention. I think that's something you could have, it could have helped as far as just the way it was organized. But overall, I think it's great research, and thank you for sharing that with us. So congratulations.
Awesome. Thank you so much. And thank you for the feedback. That's really helpful. I will consider that, let it simmer, and then make sure that I'm incorporating that for my future presentations. Thanks so much.
All right, congratulations. So I am announcing the second place winner, and I want to start by saying how much I've enjoyed hearing all of your presentations. I know you've put a lot of work into them, and you've been working on them for a long time. You've been giving it a lot of thought, and I heard in the Q&A the passion that you have around what you do, and it gives me a lot of hope for the future, honestly, and I need that. So our second place winner is Herbie Turner, and thank you so much. You did a wonderful job. Your presentation was very clear, and we particularly liked that you started with defining what the problem was. That was very, very, very helpful. So thank you for such a clear presentation. Your visuals were great as well.
All right, thank you. Yeah, I appreciate that.
So I have the privilege to announce the first place winner. I do want to thank all of the students for participating. Fantastic. We really appreciate it. You've done a wonderful job. We are so incredibly proud of each of you and what you're doing in furthering your education and graduate study, and hope you continue and go far with it. On behalf of BAMIT and with the generosity of the MIT Alumni Association, I am proud to announce that Stuart Isaacs is the first place winner. Congratulations, Stuart.
Thank you very much. Really appreciate it.
Well done. We really, really appreciated the clarity. We knew it was very clear what the problems were, what was motivating your work, and then what research has been done and where the potential areas of further investigation or finding solutions are. Wish you well. There could be some really incredible outcomes based on your research to help the people of Burkina Faso and other areas of Western Africa. So we wish you much success.
Thank you very much.
Thank you, judges, and congratulations to the winners. And just to clarify, just in case anyone missed in the beginning, there's a total of $2,300 we're giving out, and the way it's denominated is as follows. There'll be $250 going to the third place winner, $500 to the second place winner, $1,000 to the first place winner. And now I will present the People's Choice Award, and that also goes to Stuart Isaacs. Congratulations. In addition, you're welcome. In addition, all runners up will get $100 each. So thank you everyone for participating. Everyone is indeed a winner, and you can all now enact those plans you just gave us about your winnings. We'd like to again thank all of our judges and all of our competitors who volunteered themselves to be critiqued in front of an audience of alumni. We encourage you all to join us on April 16th from 3 to 4 p.m. Eastern Time for the Black Graduate Celebration. Stay tuned for an email to come soon on how to register for that. My special thanks goes to the BAMIT Programming Committee and the MIT Alumni Association staff who helped produce today's event. An archived edited video of this slam will be made available on the MIT Alumni Association YouTube channel within a week's time. Thank you very much, everyone, for participating and for attending, for the questions, for the feedback, for the presenters. This has been a very engaging event. I'm very impressed, and I think we're all very impressed with the quality of research that was shown. Thank you all, and congratulations again, and good night.